Hello everyone, welcome back to my channel. I am Isha and today we are diving into an essential topic in biotechnology, that is sterilization techniques. Let's get started. Sterilization in biotech ensures the elimination of all microbial life to maintain purity in cultures and prevent contamination. This topic is significant because sterilization is a critical process that kills microorganisms such as bacteria, viruses, fungi, and disease to prevent the spread of infection. I will cover autoclaving, dry heat sterilization, chemical sterilization, radiation, filtration, plasma sterilization, and pasteurization. First up is autoclaving. This is one of the most common methods. It uses steam under pressure to kill bacteria, viruses, and spores. Items are placed in an autoclave and heated to about 121 degrees Celsius for 15 to 20 minutes. The pressure helps to raise the boiling point of water, ensuring effective sterilization. It's perfect for sterilizing glassware, surgical instruments, and culture media. Always make sure your items are clean before autoclaving to get the best results. I will briefly explain the diagram of an autoclaving machine, which is essential for sterilizing tools and equipments. The main part is the chamber where the items are placed. The steam generator heats water to create steam, which fills the chamber. The pressure goes and temperature goes monitors the conditions to ensure they reach the required levels for effective sterilization. Lastly, the drain wall removes excess water after the process is complete. In summary, the autoclave uses high pressure steam to kill germs and ensure that instruments are safe for use. Now, I will give you a quick overview of dry heat sterilization, a method used to eliminate microorganisms. Dry heat sterilization uses hot air to kill bacteria, viruses, and spores. This method is effective for materials that can withstand high temperatures, such as glassware, metal instruments, and certain powders. The process typically involves heating the items to a temperature of 160 to 180 degrees Celsius for a specific period, usually 1 to 2 hours. One advantage of dry heat sterilization is that it does not cause corrosion, making it ideal for reusable metal tools. However, it requires longer exposure times compared to steam sterilization. In summary, dry heat sterilization is a reliable method for ensuring the safety of heat stable items. This machine works by circulating hot air to eliminate microorganisms. It is ideal for items that can withstand high temperatures like glassware and metal instruments. Inside the machine, items are placed in a chamber where hot air is maintained at temperatures typically between 160 to 180 degrees Celsius for 1 to 2 hours. One key feature is its ability to ensure thorough sterilization without causing corrosion, making it perfect for reusable tools. The machine usually includes a temperature control system to ensure the right conditions are met throughout the process. In summary, the dry sterilization machine is an effective and reliable method for sterilizing heat stable items, ensuring safety in various settings. Chemical sterilization uses specific chemicals to eliminate all microorganisms. It is especially used for heat sensitive items. Common chemicals used: ethylene oxide, which is a gas effective for delicate instruments, hydrogen peroxide, used in vapor form to kill a wide range of microbes, glutaraldehyde, a liquid that sterilizes quickly but needs careful handling. The process involves cleaning the item, exposing them to the chemical aeration to remove residue, validating effectiveness with biological indicators. Chemical sterilization is vital for ensuring safety in healthcare setting. Today I'm going to explore how hydrogen peroxide is used for chemical sterilization. Let's look at the main parts of the process. Sterilization chamber. This is where the sterilization happens. Hydrogen peroxide. This is the chemical we use for sterilizing. Instruments. These are the tools we want to clean and make safe. Now let's see what are the steps in this process. Step 1 is cleaning the instruments. First, we clean the instruments to remove any dirt or organic material. Step 2, placing in the chamber. Next, we place the cleaned instrument inside the sterilization chamber. Step 3, introducing hydrogen peroxide. Then we introduce hydrogen peroxide either in gas form or as a vapor. This chemical is effective at killing germs. Step 4, sterilization time. The instruments needed to be exposed to hydrogen peroxide for a certain amount of time, typically around 30 to 60 minutes, depending on the method. Step 5, aeration. Finally, after the exposure, we aerate the chamber to remove any leftover hydrogen peroxide, ensuring the instruments are safe to use. So, what conclusion we get from this is using hydrogen peroxide for sterilization is a great way to ensure our tools are free from harmful microorganisms. This process helps keep everything clean and safe. Radiation sterilization is a method used to eliminate microorganisms from various materials. Radiation sterilization uses high energy radiation like gamma rays or electron beams to eliminate bacteria, viruses, and fungi. It works by damaging the DNA of these microorganisms, preventing them from reproducing. The process involved is preparation. Items are cleaned to remove dirt. Packaging. They are packaged in materials that allow radiation to pass through. Exposure. The items are exposed to radiation for a specific time. Quality control. Test ensure the sterilization was effective. Advantages are it's effective for heat sensitive items that can penetrate packaging, keeping everything sealed until use. So conclusion we get here is in short, radiation sterilization is a safe and efficient way to ensure that medical supplies and food are free from harmful germs. Let's briefly explore how different type of radiation, alpha, beta, gamma, and neutrons penetrate materials. First, we have alpha particles. They can only travel a few centimeters in air and are stopped by paper or skin. They are mostly harmful if ingested or inhaled. Next is beta radiation. Beta particles can travel up to a meter in air and can be stopped by plastic or aluminium. They can penetrate skin but are less harmful from outside the body. Now, gamma rays. Gamma rays are highly penetrating and can pass through several centimeters of lead. They require dense material for shielding. Finally, neutron radiation. Neutrons can travel through meters of air and penetrate material like water and concrete. They interact with atomic nuclei, making them particularly hazardous. In summary, alpha particles are the least penetrating, followed by beta particles, while gamma rays and neutrons have the highest penetration abilities. Filtration sterilization is a way to remove germs from liquids and gases. We use special filters with very small holes to trap bacteria and other tiny organisms. So the type of filtration are depth filters, membrane filters. Depth filters are thick and catch particles inside them. Membrane filters are thin and have uniform tiny holes, great for delicate liquids. Now the question arises, where do we use it? We use filtration sterilization in growing cells in labs, making medicines to ensure they are clean, preparing vaccine and other biological products. Let's discuss about its benefits. It gets rid of bacteria and fungi, it keeps sensitive materials safe, it is quick and affordable. So there are limitations also. Limitations are it may not remove all viruses. If a liquid has a lot of particles, it might need extra filtering. So conclusion we get from here is filtration sterilization is really important in biotechnology for making safe products. Now, pointing to the diagram, at the top we start with the source liquid, which could be culture media, pharmaceuticals, or other biological fluids that can be sterilized. Before the main filtration, we may use a pre-filter to remove larger particles. This step helps protect the main filter from clogging. Next, the liquid passes through the main filter. Here, we typically have two types of filters: depth filters, membrane filters. Depth filters trap particles throughout the filter. Membrane filters have very small, uniform pores and are effective in removing bacteria. After filtering, we have the sterilized liquid, which is now free of harmful microorganisms. This can be collected for further use in experiments or production. Any particles and microorganisms that we trapped in the filter are considered waste. This is important to ensure that only clean products are used. Conclusion: this diagram shows how filtration sterilization is a simple yet effective way to ensure the safety and purity of bioproducts in biotechnology. Plasma sterilization is a modern technique used in biotechnology to eliminate harmful microorganisms. It works by using ionized gas or plasma. This plasma generates reactive particles that attack and destroy bacteria, viruses, and fungi. One of the key advantages of plasma sterilization is that it can effectively sterilize heat sensitive materials without damaging them, making it ideal for medical tools and devices. Additionally, this method leaves no toxic residue behind, making it a safe and environmentally friendly option for labs and healthcare settings. Overall, plasma sterilization is an effective way to maintain sterility and safety in biotechnology. In plasma sterilization process, first we generate plasma from a gas, often hydrogen peroxide, using energy. This ionizes the gas, creating reactive species. Next, the ionized gas fills the sterilization chamber where instruments are placed. This chamber is often vacuum sealed to enhance efficiency. As the plasma interacts with surfaces, it produces free radicals that effectively destroy microorganisms, including bacteria and viruses. After sterilization, these reactive species break down into harmless byproducts, primarily water and oxygen. Finally, we vent the chamber, returning it to normal atmospheric condition, allowing safe removal of sterilized items. Plasma sterilization is efficient, effective, and eco-friendly, making it a vital tool in biotechnology. Pasteurization, named after Louis Pasteur, is a heat treatment process that eliminates harmful microorganisms from food and beverages, particularly liquids like milk and juices. In biotech, pasteurization is often applied to enhance safety without compromising nutritional value. For instance, milk is heated to around 161 degrees Fahrenheit for 15 seconds, a method known as high temperature short time or HTST. This rapid heating kills pathogens while the quick cooling preserves flavor and nutrients, making the product safe for consumers. Biotechnology enhances pasteurization techniques, allowing for better control over temperature and time, ensuring maximum efficacy while retaining the integrity of the food. Thanks to these advancements, pasteurized products are not only safer but also have longer shelf lives, crucial for food distribution. The large jars or containers used as often referred to as pasteurization tanks or pasteurizers. These tanks are specially designed for heating and cooling food products during the pasteurization process.